like electricity goes out all the time. That is like the power plant and everything, and the electricity went out. Wow, there's nothing we can do. They're like, well, didn't you print all like handouts of all your
What?
stuff? I was like, no. And then they're like, yeah, and then they're like, no, that's what you have to do.
Wow.
So they like managed to get a generator.
A generator?
Yeah, well, the building has a generator, so like, oh, the fire generator. So if you do a presentation, we'll also have handouts.
Apparently.
Yeah, chapter.
I guess that makes sense, yeah.
Unfortunately. Okay, so I think we go, already.
Okay. No problem.
Yeah.
I understand.
Well, I was going to say, maybe we can meet up there. So, let's see here. Fall break is Thursday and Friday. But I teach Tuesday, so I would be able to go Wednesday, Thursday, or Friday.
No, we sit over here with Kate. I just don't want to talk with people.
Yeah. Just so that, you know,
Implicitly trust them.
it's just, what do you want to be?
And what person do you want to
Mostly anything. I'd love to be in packets. So she's teaching a plant biology class, but I don't know much about any of the other things that they teach. And I've been on a fellowship for my first 3 semesters here, so this will be my first TA hack, which is kind of ironic since I want to teach. That's like my goal. So really, yeah. So yeah, it's actually relevant that I can put on my resume and say, hey, I helped with this, and it is actually something that is let's see how it goes. Right? Yeah. Yeah.
I asked her, oh, there's also coffee and tea in here if anyone wants coffee or tea. Sorry, I forgot to bring it out. I'm the worst. I also don't know.
So it'd be kind of interesting to see interact with each other. We've gone at it over the same thing. I think they have some classes.
Yeah, let me know next week.
Sure.
There you'll go.
So I guess those days
And, no, doesn't matter.
probably Thursday.
Hello. It's gone out. As well.
I didn't realize it was free.
Yeah, isn't it funny when you talk to the classes in TA, like the sense of time. Yes. She's on. She hasn't got to I guess she's still going. I don't know. I know.
Guys, I'm a 5-year-old and I'm super excited about
No, it's just a horrible feedback noise, I think.
Sorry,
It sounds like microphone feedback to me.
You can switch between Earth and Mars. So if that's good, tell me how the camera is.
Remind me of what department you're in?
Anthropology?
Okay. And you, what are you studying?
Well, mostly like on global change issues in West Africa.
No, there's no one there.
Yeah.
You're mostly natural resources. Human ecologists. Households. Yep, so you feel like West Africa.
Yeah. Yeah.
You know, because similar work in Kenya.
I have muted them.
Oh, okay.
After, well, Car's not there, so. If you want to unmute them, that is your choice. Just press the red.
Someone,
I know how to use it.
someone.
Amy, yeah, I'll text Amy and ask her to just text you.
I told her, I said, wait.
Yeah, she has my number. She can text me.
Otherwise, and the screen share.
Yeah. How are you doing? Same. Yeah. Are you in town today? Yeah. How long are you there? You're on tomorrow? I'm actually covering this class. If you want to sit in on it, if you want to, I'm talking about my, well, what I study. I'd love to do that. Yeah, it's at 10:10 downstairs. So, as long as you don't make me say
You'll need to.
Right.
Shelby said.
Oh yeah, we were talking about two different projects. One is hopefully we can get paper.
It was written here.
To like hopefully submit the other exciting. Yeah. So yeah, no, it's good. And then my other project that I'm working on, I'm stuck, so I don't need to go in the lab next week. Actually, I have 10 people coming to interview me. Other things.
Well,
One's a scientist in North Carolina thing, and then one is for the research. So it'll all work out, but I don't have to like plan. I really need. Are you mostly or like when I'm in like new type of data? Oh right. I'm actually going to be in Stanford on Sunday. How do you feel about sport? Are you going to kids? Like no. Oh no, but that's the same kind of place in Stanford. Yeah. Hey, no. You did? Yeah. Okay. Oh, cool. I've never done. What's that? No, not at all. Will you leave now? No, I'm home. Yeah. We'll be down in your neighborhood. So what's really fun? Yeah. It's not far from where you live. I live in San Francisco. But maybe 3 or around.
Your bishop been exiled to the
I
park.
remember maybe I'll call for maybe we can
Well,
vibe. Say, hey, after we're done. Yeah.
Okay.
Are you guys close to? Yes, is that what you said? Like downtown? Okay, so it's a little more like rural. But we can, if you wanted to come, we just see you. Or if you wanted to be like after.
They're very small town.
Yeah, but yeah, let's be.
It's actually like right up the stairs.
Saturday night was like,
Yes, yeah.
I thought they were going to be.
our friend, my friend's parents, built this for the memorial. Saturday, so it'll be a fast. We kind of weren't thinking we were going to go.
Hi everyone, thank you so much for coming to our seminar series this week. I'm Shelby, I'm a PhD student at CE, and I'm really excited to host Dr. Caroline Tucker, who's, as most of you know, who's faculty in biology and CE. So we're really excited to have her talk about some of her research. Caroline was an undergrad in Halifax, and she studied biology at like a dual program for 2 universities, and so it was Mount St. Vincent and Dalhousie University. She then went and did her PhD at the University of Toronto in Ecology and Evolution. And then she did a postdoc at Colorado University at Boulder, and then went to France at the CNRS in Montpellier. And so I wanted to do a fun fact, so some of you might not know this, but she was, when she was an undergrad, she was an extra in a zombie movie. So definitely ask her questions about that after the seminar. Today she's talking about assessing the value of evolutionary history for conservation.
Thank you. And thank you so much, Shelby, for hosting me today, and to CE grad students for inviting me to give this talk. So it's kind of nice. The last time I was here talking about my research was for my job interview, and so some of the things I'm going to talk about, I actually told you about the ideas for at that point. So it's nice to update you on what I've done in the last 1.5 years. So I'm going to talk to you about the conservation value, if there is such a thing, for evolutionary history. And I'll start by pointing out that the 3 undeniable facts about biodiversity are: 1, that we need to consider biodiversity as a multifaceted, highly varied thing. It is not just the number of species or the rarity of species. It's something which incorporates all of the variation in life, so at many levels within, between species, and also between ecosystems. And it includes things like evolutionary history and diversity in traits and form and function. Second is that biodiversity is declining in the modern age. So this is a review from Stuart Pimentel in 2014. And you can see that for multiple groups of species, for birds, amphibians, and mammals, the number of species going extinct has just risen exponentially in the last 100 years or so. And as a response to that, there really is this call to decide what to do for this decline in biodiversity, that we need to make decisions about what we prioritize and what we don't prioritize, or what we're going to triage and what we're going to let go extinct. So this illustration was in a little blurb about my PhD supervisor who worked on some of these similar questions. And one of the things I like about it is that they only included a bunch of charismatic megafauna, which tells you a lot about what people care about. And the final fact is that despite this decline in biodiversity, we have a very incomplete understanding of how humans actually affect biodiversity and what benefits that we receive from biodiversity. So there's a nice quote from Brian Walker, who's an Australian conservationist at CSIRO. He said, "Regrettable as it might be, global biodiversity concerns will ultimately be reduced to a cost-benefit analysis." And despite that, without knowledge of the relationship between the levels of biodiversity and ecosystem function, we cannot estimate either the costs or the benefits, which is a rather sad state of affairs and a difficult place to be as a scientist. And the question of how to respond to these facts is one that lots of people have considered in different ways. So starting in the early 90s, one approach was to start thinking beyond just species. So Richard Wainwright said, if you have 2 species, how do we decide between them? To a conservationist, is a panda equivalent to a species of rat? After all, given 2 threatened taxa, it seems reasonable to give priority to the taxonomically distinct form. And so Wainwright didn't think much about branch lengths on trees or anything, but he did know that the panda is the only representative of its genus, and there are more than 50 species of rats in the genus Rattus. So, given that information, why not give some priority to panda bears? So we'll just take a short detour here to go through this concept of thinking about evolutionary history. So when we have a group of species, we can count them or describe them in lots of different ways. One of those ways is actually to consider their evolutionary history, that is, their ancestry through time. So here you can see that these species are related in what we call a phylogenetic tree. So a graph that is showing the inferred evolutionary relationships between all of these species. And this phylogeny is helpful because it provides information content. So it tells us something about the unique information associated with any given species. So for the chimpanzee and the bonobo, these are shorter branches. And then the shared information reflecting shared evolutionary history. And further, that we can put quantities or numbers to this diagram. So these branch lengths represent divergence in some way, which might be something like inferred and calibrated time, or in terms of sequence information. And therefore, as a result, we can get numbers, we can get quantities that we can use for prioritization. So the most commonly used measure is phylogenetic diversity, or PD. And it's just the sum of the branch lengths connecting a set of taxa. So here, for these three species, these charismatic circles and squares, it's just the sum of the branches here in red. You can see you get a number 8.5. And what's especially interesting about that is it gives us another way of measuring groups of species, so that you can have, for example, a set of species which is only 2 species and yet has more evolutionary history than this larger set of 3 species. And that's because we have here 2 distantly related taxa, an orangutan and a gorilla. So what might be referred to as the phylogenetic gambit is the idea that perhaps if we maximize phylogenetic diversity, PD, we might ensure that we're actually conserving a wider variety of form and function in life. And so if we choose this set of species, maybe we're getting more of something we care about, which is diversity and ecology of form and function. And this idea was formalized first and by Dan Faith in a paper in Biological Conservation, Evaluation, and Phylogenetic Diversity. And this is really the foundational paper for this entire area of research. It has more than 1,300 citations, which for ecology papers is a lot. And really, Dan has played a big role in determining the path of where this kind of work is going. And it's led to more than 1,000 papers that you can find if you search for conservation and phylogeny, which vary from those which put together lists of species in terms of their priority for preservation based on how distinctive they are in terms of their evolutionary history, which identify sites or places of higher priority for this reason, and which also extends to other areas of management, such as ecological restoration. So then it's interesting that we're now at the 25th anniversary of Dan's first paper, and yet phylogenetic diversity or evolutionary history is actually very rarely integrated into existing conservation activities. So little money is actually spent on it. I think there are really only 2 programs that I can think of that are particularly dominant. One is the EDGE program, the Zoological Society of London runs, and I'll mention that again in a little while. And the other is protected area planning in New South Wales in Australia. And almost certainly that's in part because Dan Faith is heavily involved in that. So the real question that people have been asking, though, is what are some of the reasons that leads to this disconnect between theory and application? So why is there this gap? And that was something that when I was here last time I mentioned organizing a set of workshops with my co-organizer, Arna Moores, to try and get at least from the academic side of things, what we needed to do to overcome that gap. And so I want to highlight all of these people because the work I'm talking about today will come from works that we've all been producing from these workshops, and those will be shown in the later. And so this is probably why I was so tired in the last year or so. So we had a couple meetings in Leipzig, a meeting in Ottawa, and then a meeting in London. And I think we were pretty productive. So the big question that we wanted to address was simply, why would you even want to use PD over one of the many other approaches that are out there for prioritizing biodiversity? And one of the things that is important to consider is what is the human-centric, the utility value of phylogenetic diversity. And the fact is that the literature loves to sort of mention how wonderful it is and then do little to actually test this or prove it. And so we wanted to start simply by saying, what is it that people have said phylogenetic diversity might provide to us, and then ask, is there any evidence that this is actually the case? And this, I'll take this point to note that obviously there's utility value, monetary value associated with biodiversity, but it also has intrinsic value. And so, something like the Cagato probably belongs to the museum just because it's old, amazing, and interesting. Oops. Sorry. So we come back then to this question of the phylogenetic ambit and whether or not the statement that maximizing phylogenetic diversity will ensure a wide variety of forms and functions, whether this is true. So this involved, among other things, going through this massive literature and determining what are the statements that have actually been made. They include things like perhaps it leads to greater ecosystem function or services, decreased extinctions, future speciation. And then we developed a framework, a set of relationships that we thought were sufficient to describe all of these things. And when Arna and I submitted our first application for funding, we had a little conceptual diagram that we thought was perfect and covered everything, and that was what we wanted to work off of. And then it evolved from meeting to meeting, and I think this is sort of what working groups are like. So eventually we came here. So this diagram, and I know it's complicated and I'll walk you through it piece by piece, but it basically captures the set of relationships that people have drawn in the academic literature. And it comes across, we acknowledge that these are across a wide variety of temporal scales, so from current day benefits to those in the far future, and also across spatial scales, so from local ecosystem functions all the way to global scale effects. And these are arguments for why we should care about PD, but only if they're true. So it turns out that that diagram really has three themes that we can discuss one by one. The first, and by far the most important, is that the statement that phylogenetic diversity should be a proxy or a surrogate for trait diversity. And it's the most important because you'll note that almost all of the other connections come out of this box, that trait diversity is really what it seems that we're arguing the literature matters. The second is that in some way phylogenetic diversity is tied to diversification, so either that by preserving greater phylogenetic diversity or trait diversity, you might have lowered extinction, which is probably important in the time of extinction, or you might have greater speciation into the future. And the final one is that through trait diversity, phylogenetic diversity should lead to greater ecosystem services of various types. And I'll get to that at the very end. So one of the things about this diagram is that it's saying, just go back. It's saying that essentially, if you preserve more phylogenetic diversity, you may end up with greater trait diversity, but that's obviously in comparison to something more than what. And in our case, it appears that most of the literature is really saying more than if you just chose species randomly. And so this is the baseline that we established, which is you can either select a set of species which maximize phylogenetic diversity. So that's our red line here. And you can see how much of a response variable you get. Or you can choose the same number of species, but choose them randomly with respect to PD. And that's what we show here in gray. And in this example, you can see that phylogenetic diversity is doing no better than random in terms of preserving whatever this response variable of interest is. There are, of course, also potentially the situation whereby maximizing phylogenetic diversity, you're doing better in terms of also preserving this response variable of interest. All right, so I'm going to start with the first section and the most important section, this question about surrogacy. So first, what do I mean by trait diversity? In ecology, we mean almost any variation in life. So these are the most commonly used traits in studies of plants and ecosystem function, for example. You can see great diversity across this coral reef, or diversity in size and structure across all of these keys. And we assume that these differences translate in some way to ecological differences. So here, a trait will just be any measurable feature of an organism. And that may be physiological, phenological, morphological, etc. And why is it important for PD to actually be a surrogate for traits? One reason is that trait information is often much more difficult to obtain and much less readily available in existing databases, especially when we're talking about rare species, which we often have very little data on, in comparison to the kind of genetic sequences necessary to build phylogenetic trees. The second difference, which is also really important, I think, is that we don't necessarily know what traits are important, or those traits might be very difficult to measure. And so perhaps simply by focusing on phylogenetic diversity as a way of capturing differences between organisms in general, we might be also capturing differences which are important or useful. And we also avoid the bias of choosing which traits we think are important, which in the future we may learn are not as important as we thought. So the question then to ask is, what is the expectation, at least from theory, for the relationship between phylogenetic diversity and trait diversity, or TD? And this is from Florent Mazel, who was our postdoc for this working group. And you can see that we can select species from our phylogeny, and the green species are species selected to maximize PD, and the yellow species would be an example of random selection. And here we have two traits representing our trait space. So the total amount of diversity represented by our green species is encapsulated by this triangle. You can see in this case it's actually much larger than that encapsulated by our random selection. But that's not necessarily the case. In this example, a different type of tree, a different model, you can see that our selection in green meant to maximize phylogenetic diversity actually captures less trait space than our random choice. And we can measure the surrogacy value based on some description of this. And theoretically, then, we know it's possible to have almost any relationship between trait diversity and phylogenetic diversity. So here, the surrogacy metric, if it's blue, suggests that PD is doing better than random. If it's white, it's doing as good as random. And if it's red, it's doing worse. And you can see that for at least some types of trees, types of models, it's possible that PD is actually not a good surrogate at all for trait diversity. But on the other hand, in many cases, it is, in fact, it's performing reasonably well as a surrogate. So to extend on this, one of the things to consider is that rarely are we interested in just two traits. Ecologists, at least, are really interested in multidimensional trait differences between organisms. So we define it in terms of trait space in 3 or more dimensions often. So we're interested in measuring many things on this flower, not just 2 things. And one of the reasons for this is that this conceptualization of the niche as a multidimensional space, so this idea that species occupy, their niches can be measured by many different variables, that may map onto this multidimensional trait space. That those traits may relate to fitness in a given environment, so that traits may be one way of capturing species niches. And so to expand on Florent's results, we asked, when we consider many traits, and also only for the most common models of evolution, what's our expectation for this relationship between phylogenetic diversity and trait diversity? And so this, we consider two models of evolution, which I'll discuss on the next slide. We measure trait diversity using basically the multidimensional volume or convex hull, and we consider between 2 and 12 traits. So the two models that we consider are Brownian motion, which is here, so trait values change through time by a random walk, which, as you might expect from something like genetic drift, and the Ornstein-Uhlenbeck, where we have directional selection, so towards the optimal trait value. And what we find here, so this is Brownian motion in this plot, this is the U, this is phylogenetic diversity, this is trait diversity, that as we go from few traits to many traits, in general, the relationship becomes stronger and more positive, although obviously there's a lot of variation expected, which is not surprising. And that for the OU, of course, it's saturating, but that's what we use to max the amount of additional trait diversity you're going to capture with phylogenetic diversity. So we want to know, though, particularly in relationship to random, what this means. And what we show here, so this is the number of traits on the x-axis, and on the y-axis is the r squared and the amount of variation in trait diversity being explained. That the model in which PD is being used to predict TD is actually much better than random, as long as you have more than about 2 traits. And that's true for both of these models of evolution. So this is still theoretical simulations. What does the real world actually look like? What are the models of evolution that we see in the real world? And so to do this, we took more of a big data approach. So for different geographical areas, we asked, does selecting species to maximize phylogenetic diversity do a better job of capturing trait diversity? And so we looked at mammals, birds, and coral reef fish. I'm just going to present the mammals and the birds here. We used existing phylogenies, distributional maps for the entire world, and trait databases. So this is for the mammals, which is just under 5,000 species. And this is our surrogacy metric. Basically, any value greater than 0 means that PD is a better surrogate than random. You see that we capture about 26% more functional diversity or trait diversity using phylogenetic diversity than you would get on random. And you can also see it mapped over the globe. You can see that in many cases where it's blue, we're getting really good surrogacy value. But there are certainly places here, the Sahara, New Guinea, where performance is pretty bad. For the birds, we get pretty similar results in terms of the overall performance of the surrogacy metric. So it's on average much greater than 0, and we get about 19% more trait diversity. And again, there's a fair amount of geographic variation in terms of where performance is best or worse. So we can talk about this. We can talk about when we don't have good surrogacy. A lot of these pink locations are one of 2 extreme examples. So one is where you have 2 relatively closely related species who are quite divergent in terms of their actual traits. So for example, in the Sahara, where we have some overlap with the cheetah and the sandcat, because they differ a lot in terms of body size, etc. And the other is where you have cases of really strong niche conservatism. So even though you have very deep divergences between 2 species, they maintain very similar forms and functions. This, for example, the pocket gophers, they're all different species, but they're quite similar, even though they have a very deep divergence. Still, to end this section, I think I came out of it with more confidence than I went into it with, in terms of the fact that on average at least, which is maybe all we can ask for conservation, PD is better than just randomly selecting species if we care about trait diversity. So at least the most common models of trait evolution suggest this. And for the empirical data, it actually appears to support that conclusion as well. So for almost 90% of the grid cells for the Earth, the surrogacy metric was 0 or greater. And just to finish this section by noting that, so if this line here, the dashed line, the dotted line, sorry, is your random selection of species, and this is trait diversity, and this line is selection to maximize phylogenetic diversity. you'll notice that there's still lots of space between the total amount of functional diversity that's being captured by that set than you're getting by just maximizing PD. And that's basically the unknown. That's all of the additional factors which determine what species are found in a given place on Earth, including ecological assembly mechanisms, which are likely very important in leading to non-random selections of species. And so that's still a really important area of future study. So the second section, the second set of links that we wanted to test or ask about is this idea that somehow phylogenetic diversity affects the gain or the loss of biodiversity. And so one argument is that by selecting or preserving more phylogenetic diversity might lead to lower extinctions. And there's really no evidence for that at all. I think the literature is very mixed in terms of whether you have any expectation that old groups of species are more or less likely to go extinct than young. And the data to ask about whether trait diversity is associated with extinction risk is not really available, just because of the timelines over which extinctions need to be considered. So we had to leave this sort of currently untestable. However, we could answer this second idea, which is often called evolutionary potential. That is, by preserving more phylogenetic diversity, perhaps we create a safety net that we're capturing more of the raw materials necessary for the production of biodiversity in the future. And this is something that was put forth by Dan Day from the very first paper on PD, and which has often been repeated, but certainly never been tested. So even though we know that current conservation actions are really about preserving current biodiversity, we're having effects on biodiversity over much longer timescales, and so perhaps this is something worthy of at least considering. So we asked them, do phylogenetically diverse sets of species differ in their potential for speciation? And that seems like that would be perhaps difficult to test, but we decided to do sort of a what-if question, and to say, what if we were able to go back into the deep past and make choices about which species we were actually going to save? If we had selected a more diverse, phylogenetically diverse group of species, would that have led to more species in the future than just some random selection? And so we were lucky enough to have a number of paleontologists join us at some of these meetings who know far more about these things than I do, especially one Kenzo Piedra, who's leading this paper. And so one of the things those people brought was trees which include fossils of extinct species, so that we have an idea of not only what species are currently living in those plates, but also what species there were at past time points. And so that includes the foraminifera, horses, canids, dinosaurs, and ruminants. So we got to do a little dinosaur conservation. And so the phylogenetic trees look something like this. So you can see that, for example, many of these branches just go extinct in the past. And there are, for example, the ruminants are quite diverse in the current time periods. This 0 is actually the KPG when they all went extinct for the dinosaurs. And so how do you ask this kind of question? So what we did was, so looking at these trees, we could go to a decision horizon, that is at some point in the past, and we could select, for example, some number of taxa to maximize phylogenetic diversity. Then we could ask, if we went forward to the present day, would we have, by projecting these taxa from some imminent extinction event, would we have ended up with more species than if we had just randomly selected species? And so in this case, it looks like there are 3 extant species, so we did okay. We could also select randomly, so let's say these ones, and oh, we would have done even better, it looks like. And so what this looks like then, on these plots, this is for the ruminants, so this is the point in the past, the dashed line at which you have to make your decision. Am I going to protect these animals? This is the amount of diversity that has been present before that. So in red is what would have happened if you had chosen to maximize phylogenetic diversity, and in blue is what would have happened if you had simply randomly selected species. And because there are often multiple sets of maximizing PD, and because we chose many random sets, you can see that we have confidence intervals are shown with the translation colors. So here it looks like, coming to the present day, we actually would have done better if we had selected to maximize PE at this point in the past. But, you know, if we looked a few million years later, it looks like it doesn't really matter which way you had chosen. Those lines are on top of each other. Oh, and if you looked a little bit further again, it looks like random is actually much better. And so you can see that you can do this throughout the past, and in this case, it looks like these two lines don't really differ very much. So this is for the canids. And we can do this again for the dinosaurs and for all of the other taxa that we had. So for dinosaurs, it looks like maybe a little bit better for PE-max in terms of the lineages that are protected, but not significant. And of course, they're all going to go extinct anyway, so it doesn't really matter. So then across all of these trees that we looked at, this again is a surrogacy metric, so the performance of PE in comparison to random. You see that it's very much around or even less than 0. And almost certainly there's no expectation that older lineages should be speciating more than we expect just on random. That this idea of evolutionary potential is probably not a particularly useful one. And it's worse under conditions that we frequently observe on those trees, which is when trees are more balanced, it behaves poorly, and you can see that very often trees are imbalanced, they're asymmetrical. When there's variation in diversification rates, it's quite high, and when there's higher variation in branch lengths. And so it's not really surprising that PD is not a great strategy. So not only is there little to no evidence for evolutionary potential, but the timescales at which it matters are probably irrelevant for almost any conservation program today. No one is thinking what's going to happen 100 million years from now. For very good reasons, I think. So that leads us to the final theme in this diagram, which is this idea that through trait diversity, we're associated with greater ecosystem provision and services. And for this section, I'll just note that lots of other groups are really interested in whether or not trait diversity is linked to ecosystem services. Most notably, this global intergovernmental panel, IPBES, which asks specifically what are the ecosystem services that matter to humans and how are they distributed over the planet. And we know with great confidence that species traits are related to the functions of ecosystems, so that they drive nutrient cycling, decomposition, productivity, and the transformation and acquisition of resources. We know all of these things, although there's certainly uncertainty in terms of specifics of the mechanisms. And there's a fair amount of evidence, at least experimentally, that having greater functional diversity or trait diversity tends to lead to greater ecosystem functions, at least some types of like greater primary biomass production, etc. So it seems reasonable. It seems reasonable to conclude that having greater trait diversity is likely to be tied to having greater ecosystem functions, particularly at larger scales. And there's some evidence as well that by maximizing phylogenetic diversity or trait diversity, you also are protecting useful plants to humans, so that's an ecosystem service. In this case, Phylogeny did a study in the Floristic region of South Africa, and he looked at all of the plant species that were present and categorized them in terms of the economic values they had, whether they were useful for medicine or clothing or other sources, and found that at least for some numbers of species, if you maximize the numbers, your species choice based on phylogenetic diversity, you're actually getting more useful species than if you were just randomly selecting them, as you see right. And that's often because the traits that we're interested in are clustered in particular groups of species. And so by maximizing phylogenetic diversity, we make sure we sample those. But unfortunately, all of this being true, often some degree of how money is spent comes down to what people care about, or human preference. And it's worth noting that there's actually some evidence that people care at least a little bit about some of these concepts. So the EDGE program, which is run out of the Zoological Society of London, is focused on preserving evolutionarily distinct species who are also globally endangered. And they highlight particular species which are very strange or interesting or unusual and have been very successful in bringing in funding for this purpose. So, for example, this is the purple frog from India, who's one of their top amphibians. The other is that, and there are actually studies showing that, for example, humans prefer more diverse landscapes and find them more aesthetic. So here, for example, Troubaud et al. Did a study in the Mediterranean Sea and found that they took pictures and showed people pictures with either more trait diverse or less trait diverse landscapes and had them rate them. And, you know, in fact, as trait diversity increases, people are more likely to give it a higher score. And there are also a few studies showing that humans choose, on average, more diverse in terms of traits and form and function species when they garden than in the surrounding landscapes. And also that if you give people plants and ask them to build their own little plot, they will almost always choose more diverse sets of species than you can see naturally. But on the other hand, it's true that often what we care about is specific traits, and that those traits may be present in particular clades. So that doesn't necessarily mean that humans care about maximizing phylogenetic diversity or trait diversity. It means that they like large furry mammals that are cute. So to come back to this central question and diagram about why should we care about phylogenetic diversity, I think it's worth saying that for me, if the phylogenetic ambit is about maximizing a wide variety of forms and function, this evidence that PD is not a bad surrogate for trait diversity, certainly better than random, and in the face of uncertainty that might be a good thing, is that this may be a useful approach to deal with our uncertainty in terms of what traits are important and in terms of trait values that we actually know or don't know. And that many conservation activities will prioritize utility values, so whether that's functioning ecosystems, erosion control, water, and that traits are what drive those utilities. And so if this is made that very clear in their global program. And so at the very least, I think that phylogenetic diversity is a additional component that could be considered in conservation programs, not necessarily singularly or on its own, but in concert with other approaches to prioritization, it has some value. But you know, it's worth concluding. So Rich Grenier is one of the members of our this workshop, and an email yesterday he sent. We were talking about this whole problem, and his idea, or his comments on how scientific findings make their way into conservation is the transfer of ideas and results is indirect and osmotic at best. And you know, I think that is true, unfortunately, and that's sort of where you sometimes have to leave it, I guess. And with that, again, to mention all of my co-authors and collaborators, and I'd be happy to answer questions.
So I can think of one immediate application where the botanical gardens of the world have these goals for putting X% of the world's species in the seed banks. Our own North Carolina Southeastern Seed Bank is here in the form of Michael Kuntz, who does it down at the garden. And you could say, given the right phylogeny, you could measure the phylogenetic diversity contained in the seed bank in addition to what, you know, right now it's species diversity. So I can see that happening, and it might be a kind of a fun exercise. But it sort of feels like this is a kind of seed banking problem or zoo problem in the sense of very targeted to individual animals. And zoos across the world, similarly to gardens, were sort of using phylogenetic diversity as a way of prioritizing. I can see that happening, but how do you think it relates to conservation in nature in terms of, wild habitat?
Yeah, so I mean, it's no different than choosing sites in part based on the total species richness of the site. You're just using a different metric of total diversity. And there are lots and lots, like you can use any conservation planning software with phylogenetic diversity, and there are lots of maps saying, these are the global hotspots. So all of those things are completely possible. I think it's at the scale where people don't really care about total diversity, but they have much more specific goals and limitations that it becomes sort of less applied. Yeah.
Building that, almost last thing you said, have you explored to what degree prioritizing areas of conservation based on the versus home versus would lead to conserving different areas? I feel like it depends on the spatial scale of that.
Yeah, so there's a.
Because globally will be put it some degree.
Yeah, so there is a good paper from Laura Pollock that was in Nature of Science a few months ago that shows, so it's definitely different areas. You can consider what new areas do I need to add to the existing species richness based sites, and it's not terrible. It's like 15% different, but it's definitely different. So especially trait diversity. So they also looked at trait diversity. If you know trait diversity, those are really different areas than where species are most rich.
So thinking about the second test, the second speciation extinction is, and it doesn't seem like it's a fair test to me. I mean, if the species numbers are constant, there's always going to be lineages that no matter what's changing. Is there a way to define that differently that is more fair to the idea?
So if the background rate is fixed, then if you choose a random set or a non-random set, should it be?
And is the idea strictly with respect to species numbers? Which is what that test is.
Right.
That's the way to do it. Of course, if the species numbers are constant, then they can't be more perfect.
So it's entirely some measure of species based. They have a lot of lineage through time, lots of them through the area under the curve as well. But yeah, I'm actually not sure. I think so. For me, those fossil trees come with their own set of sort of uncertainties and concerns, anyways.
Yeah.
So yeah, like I would be. I don't know. So I don't know about that. I think they would be unlikely that they don't show PD is important, and that it's unlikely that any of the biases would make it important to give control for them, but I'm not sure. And I'll pass that on. Yeah.
Oh, because it may be conflated with trait in the same sense that you have really distinct lineages, really distinct traits.
And that's the thing we know nothing about that. Yeah. Right. And that's the missing bit, and that could be really important and interesting, but we don't know anything about those traits. Yeah.
There's lots of stuff to think about. Stimulating thought. But one thing I was sitting and thinking about, and to some extent, you're testing the phylogenetic diversity criterion for conservation as opposed to random species, which is basically sort of a species, both of them are sort of species-oriented conservation priorities. And I was wondering whether you had considered in your working groups whether there were other sort of synthetic ways to prioritizing conservation. I can think of one, there may be others. You could conserve as many species as possible randomly or without diversity. But suppose you did something like decided you were just going to conserve ecosystems.
Right.
How many species would you get then?
Yep.
Or something like that. And then maybe there are other ways of doing it. Maybe it would be just geographic areas of the world, equal representation.
Yeah, and I probably would have said conservative ecosystems is a more reasonable and like better approach.
Is there some way you can compare them though to as strategies?
I mean, it becomes a little harder because you can put a firm number on phylogenetic diversity and species number, and like ecosystem is obviously a bit more vague around the edges.
Possibly. Is it easier to classify species than it is ecosystems? Certainly.
Right, right.
There's been a lot of stages.
No, that's a good point.
And there may be more in the next one. They're both hierarchical and you can slice them at different levels.
Yeah.
So,
Yeah, and I think a more useful approach would be to combine these things anyways, right? It's ecosystems and phylogenetic diversity or species.
for a lot of it.
Well, I was just wondering, kind of based on that same question, that we just looked at contemporary lists of endangered species in current protected areas and conservation areas. Are we doing a very good job for phylogenetic diversity?
Yeah, so what I do know based on a paper that we just put out was for coral reef fish. In fact, where we do the worst is the no data species. So they tend to be the most distinct, and we also know the least about them. It's hard to say otherwise. There's a lot of variation in terms of the other categories that you could be really, evolutionary distinct in an endangered category or a non-threatened category. So there's no clear trend otherwise. Yeah.
Yeah, for a lot of at least the second section, you focused on certain clades or taxa. What about functional diversity of species that co-evolved, say like bees and plants or something like that? How would you consider those very different clades, but they need each other to survive?
Yeah, so this sort of thing is almost always in a single trophic level, precisely for that reason. And this is another problem, is that when you have very different phylogenies, they tend to be very different in ages. So the phylogeny of birds is just going to be much older than one for some other, like mammals or something. And how to combine things, people use all sorts of different approaches. They just take the mode, like they take the extreme values or something, try and say, well, we'll protect the most diverse birds and the most diverse, mammals, but they don't think about, but some of those mammals evolved with some of those parasites or something. I don't think that happens that I know of.
That was great, Carolyn. So towards the end of the first part, you ended the first part with the sort of theoretical curve showing, okay, here's what was protected at random, here's what maximizing PD does, and here's the actual sort of maximization of trait diversity. And I'm just thinking, you must have, for those groups that you guys looked at for the mammals, you must be able to say roughly on average what fraction of that maximal trait diversity you actually are capturing with the maximizing of PD. And so do you have a sense of that off your head?
Yeah, so I think there are two answers. One is that actually it often doesn't take very many species to sort of maximize your functional diversity or trait diversity, because once you've selected, one predator and one whatever, you pretty much cover the space. So it doesn't necessarily take more than like 30% of the whole set of species present in a site. There's lots of redundancy. That's definitely true. I think that those trait databases are very small, so we only had like 4 to 6 traits. So if you had 10 traits, maybe that wouldn't be true. And I think that's probably the case. We just don't have the data to ask that.
But more so, well, I guess what I'm thinking about is not how many species it takes to capture it all, but just for controlling for a number of species, which is what you're doing on sort of grid cell basis, right?
Yeah.
Controlling for that, what is the trait diversity when you maximize PD relative to if you had, you actually take the, just like in your initial cartoon diagram, you actually maximize trait diversity. What's the proportion?
Yeah. I think it's actually really low. I think that, so the values are like 0.29 or something. That's of the total.
Well, that is.
Yes. Yes.
Oh, sorry.
Yeah. So like 29%. Yeah. It's not. There's a lot of missing information. I think that the translating step from what the
biogeographic species pool looks like to what your grid cell looks like is the one where we don't have a lot of information.
Well, thank you everyone for coming, and thank you, Caroline, for that awesome talk. I'm sure Caroline would love to answer any more questions. We're gonna head over to Top of the Hill for some adult beverages if anybody would like to join. Also, just a shout out for our next seminar in 2 weeks. We're having Dr. Alina Kant, who's coming from UNC-CH to talk about coral reef ecology. So, thank you.
This one's great.
Yeah. Yeah. Great.
Colin, why don't you send me an email
Yeah, I
if you
will.
go next week?
Yeah, I think it's most likely.
So I have this.
So you,
Yeah. That was legit. Sweet. Way to go. That's budget friendly. Look at that. Yeah. If you have a curve. Yeah.
Yeah.
Right. Yeah. Right? I think so. Yeah, and I think that curve,
That's your decay.
yeah, and I think
You want to inventory of the amount of sediment lost.
right, right,
Maybe you do that as what your initial minus was left.
yeah,
Area integrated. So it's a by difference calculation. After you integrate what's left, it's a by difference calculation.
yeah, because we know what we put in, right, and we know what we're measuring. So,
And is it that one? That one?
yeah.
Figure that out. and let me know.
Yeah, definitely, right? I'm gonna work on the outline stuff tomorrow, and I'll have an updated one to show in lab group just so you can scan over it and make sure it's nice and clear. That's cool. And then I'll work on Monday, Tuesday, question because I think that's driving what to do next with the project. And I think there's this Mary Leibhardt wants to come by and take pictures, but she can come by the lab and sectioning sediment is perfect.
Yeah, it'll be a follow-up.
Yeah, and I think I wanted to do another clay calibration curve, so she can come with us and do that. So that's perfect. It will work out really well, and I feel a lot better about not rushing into the lab.
Yeah, I don't want you to go there.
Like it's time consuming, not the running experiments, but it's set up and the sand removal gets so
Well, it's a lot of
and knowing yes. Yeah. All right, well yeah. I'll see you Monday. Have a great weekend. All right, I'll see you on Monday. Me too. Okay. Yeah,
I grab my stuff.
You're gonna
Yeah, I'll just tell Caroline won't leave it in the lobby.
I think Brian's okay. We can give him back the clicker.
Oh, yes.
Actually, I'll do that. It's actually me tomorrow.
Yeah, ask him if he wants to figure out how to, I don't know how to draw.
Oh, I do. Okay. I'm just gonna push on the
Well, I don't think it was moved in the
Yeah.
Yeah.
before. Unless they replace all those.
Oh, yeah, just stop recording.